Hello everyone, welcome to the Biotech School. Today we are going to learn about the gluconeogenesis. So, what do you mean by gluconeogenesis? Gluconeogenesis is the synthesis of glucose from non carbohydrate sources. So, this is a universal pathway. It is found in all animals, plants, fungi, and microorganisms. So, the site for gluconeogenesis is liver and kidney. Liver is responsible for 85 to 95% of the glucose that is made. But during starvation or during metabolic acidosis, the kidney is capable of making glucose and they may contribute up to 50% of the glucose that is formed. So generally liver is the organ which is responsible for glucose formation, but sometimes during starvation or during metabolic acidosis kidney can also contribute for the formation of glucose. There are some precursors to gluconeogenesis pathway. First is glycolytic products, which are like lactate, pyruvate, and glycerol. Then there are citric acid cycle intermediates and some amino acid except lysine and leucine. Lysine and leucine do not take part and do not form precursor in the gluconeogenetic cycle. So the animal cell carry out gluconeogenesis from 3 carbon and 4 carbon precursors, but there is no way to convert acetyl coenzyme A, which is a 2 carbon precursor, to pyruvate and oxaloacetate. So that is why they carry out gluconeogenesis from 3 carbon and 4 carbon precursor. Acetyl coenzyme A is a 2 carbon precursor. So fatty acid and lysine and leucine are not substrate for gluconeogenesis because most fatty acids yield only acetyl coenzyme A upon degradation. So fatty acid and lysine leucine are not substrate for gluconeogenesis because most fatty acid yields only acetyl coenzyme A upon degradation. Some plants and microbes can convert acetyl coenzyme A to glucose. Conversion of pyruvate to phosphoenol pyruvate. So pyruvate, when combined with pyruvate carboxylase, this is the enzyme, with the help of carbon dioxide and ATP, then oxaloacetate is created. The pyruvate, and to know about the pyruvate carboxylase, it is a mitochondrial allosteric enzyme with the prosthetic group of biotin and also a CO2 carrier. It requires Mn2 plus and Mg2 plus for its activity, and the site of its action is matrix of mitochondria. So acetyl CoA acts an allosteric activator of pyruvate. The oxaloacetate is then converted to phosphoenol pyruvate and carbon dioxide through the enzyme phosphoenol pyruvate carboxykinase with the help of GTP and 1Mn2 plus. So to know about this enzyme phosphoenol pyruvate carboxykinase, it is found in both mitochondria and cytosol. In some organism it is found in mitochondria and for some organism it is found in cytosol. So in chicken, pigeons and rabbit it is found in mitochondria, and in rat and mouse in their liver it is found in the cytosol. In some cases, and these kind of cases where PP carboxykinase is found in the cytosol, In that case, the oxaloacetate cannot cross the mitochondrial membrane because the mitochondrial membrane that has no transporter for oxaloacetate. It do not have a transporter for oxaloacetate. So what they do in that case is it becomes malate leaves the mitochondria. The oxaloacetate is converted into malate through a reduction of a NADH, and once it reaches inside the mitochondria, the malate leaves the mitochondria and comes to cytosol in the form of with the adaptation of NAD from and becomes NADH and forms oxaloacetate in the cytosol. So here is the overall process of gluconeogenesis, where the pyruvate is converted to phosphoenol pyruvate, and through the process it convert to fructose 1,6 bisphosphate, where there is an enzyme known as fructose 1,6 bisphosphatase, that is an allosterically regulated enzyme, where it is activated by the enzyme citrate, and it is inhibited by fructose 2,6 bisphosphate and also AMP. Then from fructose 1,6 bisphosphate it is get converted to fructose 6 phosphate, which then get converted into glucose 6 phosphate, and the glucose 6 phosphate it occurs in the endoplasmic reticulum of liver, and the glucose 6 phosphate it occurs in liver and kidney, but not in muscles and brain. So this enzyme is absent in muscle and brain, so it is not synthesized in muscle and brain. Glucose 6 phosphate is not do not get synthesized, and there is no gluconeogenesis. So this is the reason why there is no gluconeogenesis in brain and muscles. And then from glucose 6 phosphate it get converted to glucose, and the overall reaction looks like this, where 2 pyruvate plus 2 NADH plus 4 ATP plus 2 GTP plus 6 H2O get converted into glucose and 2 NAD plus 2 GTP plus 4 ATP. So there is a glucose alanine cycle which also take place in liver and muscles. When the pyruvate through gluconeogenesis get converts into glucose, then the glucose through blood goes to muscles, and there the glutamate and pyruvate reacts with each other to form at alpha ketoglutarate and alanine. This process is known as transamination. Then the alanine then transfused through blood and then again goes to the liver, and then they get converted to pyruvate, and again the gluconeogenesis takes place. There is then the regulation of gluconeogenesis. So the regulatory site is cytosol. It is same as the glycolysis. Without the metabolic control, both gluconeogenesis and glycolysis could continue without any net benefit to the cell and with considerable consumption of ATP. So when there will be glycolysis, simultaneously there will be gluconeogenesis. So this will consume a considerable amount of ATP. So it is regulated in such a way that when glycolysis is inhibited, the other is not active, and vice versa. Gluconeogenesis is controlled and regulated by allosteric and substrate level mechanism. So this is the total overall regulation of gluconeogenesis, where it is pyruvate getting converted into phosphoenol pyruvate with the enzyme acetyl coenzyme A is the activator, and the enzyme is pyruvate carboxylase. So then the through process it converted into fructose 1,6 bisphosphate, and fructose 1,6 bisphosphatase is the enzyme where it get converted to fructose 6 phosphate, where the negative inhibitors are AMP and fructose 2,6 bisphosphate, and then the fructose 6 phosphate is converted into glucose 6 phosphate, and the glucose 6 phosphate is then converted into glucose through the enzyme glucose 6 phosphatase, and this is the substrate level control.